Good afternoon, everyone. Welcome to our MHRC seminar this afternoon in Toronto. My name is David Hood, and it's a pleasure for me to introduce our speaker today, Dr. Steve Siegel from the University of Missouri. Dr. Siegel is the Curator's Distinguished Professor and Chancellor's Professor of Research Excellence in Medical Physiology and Pharmacology in the Department of Medicine, School of Medicine, University of Missouri. He has a list of accolades that's an arm length long. I'll give you a little bit of history. Steve did his master's in exercise physiology at UC Berkeley with George Brooks. Did his PhD in physiology at the University of Michigan under John Faulkner and Tim White. He did his postdoctoral fellowship at the University of Virginia with Brian Dooling. These are people that are outstanding scientists in each of these fields, and no doubt have had a strong impact on Steve and his productivity and the great work that he has done. Steve went on to Penn State, worked there as an assistant professor for 5 years, and he was at Yale University in New Haven for 14 years, and he's been at the University of Missouri for 17 years. His PubMed listing is over 150 publications in the area of blood flow regulation, microvasculature. It's just exciting to have Steve here. I remember many years ago him giving one of the most compelling talks I've ever seen at Ontario Exercise Physiology Conference in Collingwood. Some years ago, I was just captivated by Steve and the way he delivered the message on microvasculature and exercise. So I'm very excited that Steve has joined us here today. Steve, if you wouldn't mind, turn on your camera and let's have a look, and you can turn on your microphone, share your slides with us, and let's get our seminar started this afternoon.
Good afternoon, everybody. It's really a pleasure for me to join you. Dave, can you hear me okay?
We're good. Yep, excellent.
Okay. I'm going to switch over to a laser pointer for a bit. All right, so just to acknowledge our funding through the NIH, through the American Heart Association, and also through my Mulligan professorship. I am, as Dave alluded to, I'm a faculty member in the medical school here, and I'm also an investigator at the Dalton Cardiovascular Research Center. So I'm going to talk to you today about a relatively new areas of work in my lab. The history of my lab has really been studying blood flow regulation in the microcirculation. And some years ago, I decided I needed to make a career change. And so we've moved into the areas that I'll be describing today. And then I'm going to give you an overview of some of our studies. And I'll be happy to answer questions at the end. If anybody has anything really urgent, you can raise your hand during the talk, and I'll do my best to try and address it. So here we go. All right, so we're all getting a bit older. And when we think about advancing age, one of the things, at least in my world, is thinking about cardiovascular, but in specific vascular dysfunction. And of course, a big player in that is oxidative stress. And I'm not going to go through all this in detail, but oxidative stress, we've got mitochondria, we've got NADPH oxidase, et cetera. And the thing that I want to point to is there's many sources of generating, multiple sources of generating superoxide. And then, as we know, superoxide can be dismutated outside the cell in the cytoplasm and the mitochondria to form hydrogen peroxide. Hydrogen peroxide is a relatively stable ROS, reactive oxygen species. It's involved in signaling, but it is also detrimental to cell health. And two of the things that happens is we wind up, as a consequence of oxidative stress, generating nitric oxide and peroxynitrite. And this is just a brief introduction to some of the things that I'll be talking about later in the talk. Rather than oxidative stress being lethal, I want to introduce a paradigm shift, and that oxidative stress protects blood vessels from oxidative stress. And I'll develop that for you. The question, of course, is how does that happen? All right, so I want to introduce you first to some of the preparations that we'll be using, and I'll do this throughout the talk. What you're looking at here is an isolated feed artery. These vessels are about 100 microns in diameter. You're looking at it in differential interference contrast here. This is just the vessel laying on the stage after it's been partially digested by an enzymatic treatment and trituration. And what you'll see in the upper square here, you'll see smooth muscle cells wrapped around the vessel, stained for smooth muscle alpha actin. And in the bottom part of the vessel, what we've been able to do is remove the endothelial cell, or remove the smooth muscle cells and leave the endothelium intact. And you can see the nuclei. Here's a smooth muscle cell coming off. This is a blow up of the endothelium. And here it's been stained for connexin 40, which is a gap junction protein. And I'll talk about those a little later. And as you would expect, the gap junction proteins are localized around the periphery of the individual endothelial cells with a calibration bar of 50 microns here. All right, and so we developed some years ago, and there were papers, Matthew Socha was the first author on this. The methods are published in the Journal of Visualized Experiments, or JOVE. And we developed a preparation as the endothelial tube. So this is an intact endothelial tube. It's deflated, it's not pressurized. And we can study this as an entity unto itself. We've got a number of papers on this. Today, I'm just going to talk about it in the context of oxidative stress. And really, the work that got us going was this 2015 paper with Matthew. Matt is the first author. And what we're going to show here is that advanced stage protects microvascular endothelium from aberrant calcium influx and cell death induced by hydrogen peroxide. Okay, and the experiments that we're going to be doing, or did, are C57 Black 6J mice. We're going to look at young mice. 4 months corresponds to animals that are approximately mid-20s in humans. Older animals, 24 months old, approximates the age of me, mid-60s. Okay, and so mouse years are a lot faster than human years. Okay, so these are our young and old groups. And this first study is all going to be done in male animals. And what we're going to show here is that aging reduces the calcium response of microvascular endothelium to hydrogen peroxide. I won't bore you with the details, but we spent a lot of time calibrating the concentration of hydrogen peroxide. We're going to use 200 micromolar, and we're going to study the endothelial tubes slightly cooler than normal body temperature. We find that if we warm them up to 37 degrees, the preparations deteriorate. If we keep them at 32 degrees C, they're good for several hours. So these are the conditions under which we've done these experiments. Okay. And what Matt showed, and we're going to be looking at calcium concentration using a ratiometric technique. This is Fura2 in the cell. So we're looking at the ratio of 340 to 380. And an increase in this ratio shows an increase in calcium intracellularly. So we incubate the preparations with a dye, wash out excess dye, let them equilibrate, and then proceed with the experiment. Okay, so at baseline, there's no fluorescence. We expose to 200 micromolar hydrogen peroxide. And what you can see that when there is normal calcium in the extracellular fluid and the physiological saline solution, we see a progressive increase in intracellular calcium. When we wash in control PSS, the response decays. These are in young mice. When we repeat the experiment in old mice, what you'll see is there's a much lower concentration response. There's much lower increase in calcium. So aging protects the endothelium from a calcium influx. There's a lot more to this study, but I want to show you one of the key findings. And if we do a live dead cell assay, the live assay, calcium acetoxy methyl ester is taken up by cells, and live cells cleave the ester so that the living cells turn green. Ethidium bromide, like propylium iodide, is normally impermeant to the cell membrane. When cells are compromised and the membrane is disrupted, ethidium bromide can enter and then intercalate with the nucleus. And so what you see are the nuclei of dead or dying cells. And you can see already that the young have many more dead or dying cells than the old. Okay, and if we merge the images, it's exactly the same thing. Again, more dead and dying cells in young than you see in old. And this is just to show you what the endothelial tubes look like. And if we look at the percentage of ethidium bromide positive cells, okay, we're killing about 5 times as many endothelial cells in the young as compared to the old. The old are protected. And I'm not going to show you the data here, but if you look at ROS production by isolated endothelial cell tubes or in intact vessels, you see a much higher level of intrinsic ROS production in the old animals. So the idea is that a chronic exposure to increased oxidative stress somehow leads to adaptations that are protective. Okay, and just to give you the take-home message, what happens is with aging, we attenuate calcium influx. So that the protective mechanism it involves a reduction in calcium influx, as you've already seen. And of course, one of the questions is which ion channels are involved, and I will address that later in the talk. So the mechanism is a reduction, mechanism, reduced calcium influx. So that was in endothelial tubes. That's what got us going. The question then is there a difference between smooth muscle cells and endothelial cells in their resilience to oxidative stress? And so the approach then, of course, is to use an intact vessel. And the vessel that we're going to use is the superior epigastric artery. Again, the methods on this have been published. And just to show you where we are, this is the anesthetized mouse. The skin has been peeled away. And what you're looking at is the abdomen. And the superior epigastric artery runs from just underneath the xiphoid process along the abdominal wall. What we do then is we dissect out on one side. So here's the abdominal musculature. Here's the superior epigastric artery running alongside the vein. A little bit hard to see them both. The major vessel that you see here is the vein. But ultimately, we excise the vessel, clean it up, and then cannulate it on both ends, and then pressurize it to 100 centimeters of water, or about 75 millimeters mercury. So this is the preparation that we'll be studying. Again, what we have here is a monolayer of endothelial cells surrounded by a monolayer of vascular smooth muscle cells. All right, and the key work was done by Chuck Norton. You'll see him at the end of the talk. And again, another JPhys paper. And here we're showing that advanced age protects the resistance arteries of mouse skeletal muscle by attenuating apoptosis induced by hydrogen peroxide. And I'll take you through some of the key points of this study. All right, and so we're going to look at, let me see if I can move this here. Yeah. Hydrogen peroxide induced cell death. And we're going to look at smooth muscle versus endothelium. And now we're going to do the male versus female comparison as well. Oh, what happened? Okay. There's three conditions that we're going to be looking at the vessels. Control is we just have the vessel sitting in normal physiological saline solution containing 2 millimolar calcium. We're going to expose the vessels to hydrogen peroxide. And the other thing that we're going to do is denude the endothelium. And the way that we do that is we pass an air bubble through the lumen of the vessel. Okay, and I've already told you about how hydrogen peroxide can, it permeabilizes the membrane. Before we used ethidium bromide, and now we're using propylium iodide. It's the same thing. It cannot enter the cell in healthy cells. When cell membranes are compromised, it diffuses into the cell and intercalates with the cell nucleus. What you'll see is we have nuclei going in this direction. These are smooth muscle cells. They orient around the vessel, as you saw earlier, and then you'll see nuclei going in this direction. These are endothelial cells, and I'll show you more examples. So these are endothelial cells. Herx 33342 is a membrane permeant dye, and it labels the nuclei of all cells, and you can see that very nicely here. The endothelial cells are all oval and running along the vessel axis. The smooth muscle cells are running perpendicular. There's really no effect of hydrogen peroxide. These are different vessels. Hydrogen peroxide doesn't do anything to Herx staining because, as I said, it's a membrane permeant dye. And the thing that I want to point out here is with the Herx or with the denudation, what you see now are pretty much only nuclei of the smooth muscle cells. There's no endothelium. These little round balls that you see here are adventitial cells that have been, in this case, damaged during the course of dissecting the vessel. But what we're really paying attention to are the endothelial cells that look like this and the smooth muscle cells that look like this. Okay, and what we're going to do is we're going to take a region of interest in the middle of the vessel, and we're going to quantify the total number of smooth muscle cells and the total number of endothelial cells. And then we're going to look at the dead smooth muscle and dead endothelial cells relative to all cells. And again, denudation is basically showing us that killing the endothelial cells doesn't do anything. All right, and so here's the data then. If we count the number of cells in the region of interest in male and female mice, we have just under 120 smooth muscle cells. There's no sex differences here. If we look at endothelial cells, there's about the same number in each, slightly fewer than the smooth muscle cells, about almost 90. But look at the number of dead cells, okay? smooth muscle cell death is much higher than endothelial cell death in males. And look what happens in females is we have a tremendous reduction in the number of smooth muscle cells that are killed. So being female protects smooth muscle cells during exposure to ROS, or in this case, hydrogen peroxide. Endothelial cell death is much lower. They are more resilient, and it's even lower in females as compared to males. If we express this as a percentage in males and females, okay, the smooth muscle cell death is much greater in males. Females have lower smooth muscle cell death and also lower endothelial cell death. So smooth muscle cells are far more susceptible to the effects of hydrogen peroxide. And that's just recapitulated here. And the other thing is that if we now take away the endothelial cells, look what happens to smooth muscle cell death is we get about a 50% increase in the number of smooth muscle cells. And the reason for doing all of these controls is that, as I'm showing you here, denuding the endothelium in and of itself under controlled conditions with controlled PSS has no adverse effects on smooth muscle. But when we then expose the vessel to hydrogen peroxide after denudation, look at what happens to smooth muscle cell death. So to summarize, females are more resilient than males. Endothelial cells are more resilient than smooth muscle cells. And we have evidence now that the endothelium is protecting smooth muscle cells. And of course, the question that comes to mind is how, and everybody likes to think about nitric oxide. Well, it's not quite that simple. All right, now we're going to look at smooth muscle cell death preparations under control conditions. You've already seen this. These experiments are all in males because, as you saw earlier, they have a higher susceptibility of smooth muscle cell death. If we expose the preparation to L-name, instead of L-name is a nitric oxide, it inhibits nitric oxide synthase. So we're blocking NO production. Instead of so when we inhibit nitric oxide production, we are reducing the number of smooth muscle cells. So somehow nitric oxide is detrimental to smooth muscle cells. And FedUps, longer name, we just call it FedUps, is a peroxynitrite scavenger. And when we scavenge peroxynitrite, we also attenuate smooth muscle cell death. And so the idea is that nitric oxide and superoxide form peroxynitrite. Okay, and so by scavenging nitric oxide, we eliminate a precursor for peroxynitrite. And by scavenging peroxynitrite, we attenuate cell death as well. Okay, if we look at endothelial cell death, as you've seen already, it's much lower. It's not affected by L-name, but it goes essentially to zero when we scavenge peroxynitrite. So nitric oxide is not, it's not protective. People generally think of NO, a vasodilator, et cetera, and that does good things. In this case, because it's interacting with superoxide, it winds up being a bad guy under these conditions. Okay. Another way that smooth muscle and endothelial cells interact with each other is through gap junctions. Myoendothelial coupling is a term that probably many of you have heard. Carboxylone is a gap junction inhibitor. And when we inhibit gap junctions, in fact, we're increasing smooth muscle cell death by about 20%. So this would suggest that myoendothelial coupling is somehow protective to smooth muscle cells. And when we disrupt that, when we dissociate smooth muscle cells from endothelial cells by inhibiting gap junctions, they become more receptive to being killed by hydrogen peroxide. And the endothelium is unaffected by treatment with carboxylone. So this is just addressing two common signaling pathways between the endothelium and smooth muscle. NO is bad, gap junctions are good under the conditions of these experiments. Okay, and now NO and peroxynitrite augment calcium, hydrogen peroxide induced calcium entry. And you can see that here again, we're looking at intracellular calcium under controlled conditions during exposure to hydrogen peroxide. Oh, and what I'll tell you is in the intact vessel, we find that we can go longer. We can use a longer exposure to hydrogen peroxide. So we're typically going for 50 minutes. That's 50, and you can see that here. So here's the progressive increase in intracellular calcium. If we inhibit nitric oxide with L-name, or if we inhibit peroxynitrite with FedUps, what you can see is we attenuate calcium entry under both conditions. And I don't know why I'm having a problem advancing the slides, but I will try again. Let's try it this way. We'll do it this way. Okay, and if we remove extracellular calcium from the bath, what you can see is, well, during control conditions, here's the increase in intracellular calcium, falls when we wash. In the absence of extracellular calcium, there's no rise. And this tells you that all of the calcium that we see, all of the increase that we see during exposure is coming from extracellular influx. All right, and so if we look at smooth muscle cell death in young versus old, you've seen this already. And in denuded, the difference between young and old persists. But when we denude the vessel, you'll see that, as I've shown you already, this is just summary data. There's an increase in smooth muscle cell death and in both the intact and the denuded vessels, but the difference between young and old persists. Endothelial cell death, low to begin with, and again, slightly attenuated with aging. And this is young intact and old intact. And this is just to reiterate the fact that aging radically attenuates the rise in intracellular calcium during hydrogen peroxide exposure. So the endothelium protects smooth muscle cells from hydrogen peroxide induced calcium entry. And I will develop that a bit more later in the talk. All right, so how does smooth muscle cell death occur? And what I'm going to show you in this next series of slides is that it's happening through intrinsic apoptosis. Some of you may be familiar with the idea that calcium overload, it triggers cytochrome C release from the mitochondria, and that is a major trigger for the intrinsic pathway of apoptosis. And so here in the first two slides, panels, we're looking at mitotracker, which just shows you where the mitochondria are. Under control conditions, the mitochondria are red. We're labeling the cell nucleus with Herx dye again. After exposure to hydrogen peroxide, what you can see is the mitochondria are essentially disrupted. Okay, and now we're immunostaining for cytochrome C, and the cytochrome C under control conditions is pretty much localizing within the mitochondria, as you see. After exposure to hydrogen peroxide, it has basically escaped the mitochondria. There's only a few that are remaining intact. In this case, TOPRO is being used to label cell nuclei. Okay, caspases are integral. Activation of caspase is integral to apoptosis. Once you activate caspases, here I'm showing caspase 9, and here's caspase 3. Both of these, when they become active, are proteolytic enzymes, and they just start chewing up proteins inside the cell. And what you can see is there's very little cleaved caspase 9 or very little cleaved caspase 3 under control conditions. But after exposure to hydrogen peroxide, you can see that both of these catalytic enzymes really light up. So what we're doing by increasing intracellular calcium, much more so in young versus old, is that we are activating intrinsic apoptosis subsequent to calcium overload. Okay, and then another marker of apoptosis is staining for an Annexin-5. And I'm showing you now, this is one of my favorite pictures. Here you can see, here's a dead smooth muscle cell nucleus. Here's a live smooth muscle cell nucleus. Okay, and then the endothelial cells, in this case, phosphatidylserine, phosphatidylcholine is a membrane lipid. I'm sorry, that's phosphatidylcholine. And normally it is facing on the inner surface of the lipid bilayer on the cell membrane. But when cells are undergoing apoptosis, it flips. And now the phosphatidylcholine becomes exposed to the extracellular domain, and it will, if you put an Annexin-5 in the bath, it will stain for an Annexin-5. So what you're seeing are endothelial cells that are undergoing apoptosis. Some are just beginning, some are well along the way. And if you look at each of these, you can see the nucleus is labeled with propyridium iodide. So it's just an independent marker showing that cells are undergoing apoptosis as a consequence of exposure to hydrogen peroxide. And I apologize if I sound like I'm a little bit jumping around, but it's so strange to be staring only at my screen when I'm used to interacting with an audience. So this is one of the setbacks, if you will, of doing a seminar by Zoom. And it's just a bit strange. So please bear with me, and I'll continue to do my best. So do vessels from different organs respond to hydrogen peroxide in a similar manner? Okay, and the next place we looked, and I'll say this is largely driven by Chuck's interest, is the posterior cerebral artery, which sits right about here at the base of the brain. Okay, and this was a paper that came out earlier this year in ACTA, and what it's going to reiterate some of what you've seen, but we're going to address mechanism a bit more. Endothelial cells promote smooth muscle cell resilience. They're protective to hydrogen peroxide-induced cell death in mouse cerebral arteries. Okay, and we're going to do this in males because, as you've seen earlier, males have a higher preponderance for smooth muscle cell death during hydrogen peroxide exposure. Okay, again, we're going to use 200 micromolar, same duration, same temperature. And really, we're just looking to see if vessels from the brain behave in a manner similar to those from skeletal muscle. Okay, and you've seen this already. Hydrogen peroxide, and then control is just physiological saline solution with 2 millimolar calcium. Here we're staining with the Herx 33342 to show all nuclei. Here we're staining with propyridium iodide. And under control conditions, once again, the only cells that you see damaged, these little round dots, are adventitial cells that are damaged during the process of dissection. But there are no either smooth muscle or endothelial cells damaged, as you can see here, when we merge the image, is by the process of removing the vessels. Whereas those that have been exposed to hydrogen peroxide, it's the same thing that you've seen before. In this case, the yellow arrow is pointing to a smooth muscle cell nucleus. A white arrow is pointing to an endothelial cell nucleus, okay? And again, here's our region of interest in the calibration bar. And so these pretty yellow boxes are the way that ACTA wants you to present your data. So we're going to see some data slides that look like this. As you've seen already, the smooth muscle cell, here's in the presence of endothelial cells. And when we denude the endothelium, we see an increase in cell death. Okay. Now, you may or may not remember, but what we're seeing now, a big difference between skeletal muscle is that normally smooth muscle cell death would be up here in the presence of the endothelium, and smooth muscle cell death would be up here in the absence of the endothelium in skeletal muscle. For some reason, the brain vasculature is more resilient to oxidative stress. And going into these studies, I would have expected that the brain would be more susceptible to oxidative stress. And since we're talking about the brain, this is where 200 micromolar comes from. There's one study that actually measured quantitatively the amount of hydrogen peroxide being formed during ischemia reperfusion in the rat brain. And that's where this number 200 micromolar came from. I'll tell you that when we work with higher concentrations, we wind up killing all the cells in a much shorter period of time. And if we work with lower concentrations of hydrogen peroxide, you can go more than an hour, sometimes an hour and a half, and you still don't see a lot of cell death. And by the 2 hours, 2.5 hours, you're worrying about the integrity of your preparation. So that's just a little bit of boundary conditions for how we arrived at this concentration and this duration of hydrogen peroxide. ZVAD it's one of those long tongue twisters. It is a non-selective inhibitor of caspase enzymes. So we're going to prevent the catalytic activity of these enzymes when we add ZVAD to the solution. These are separate experiments now. And so here is smooth muscle cell death when endothelium are present. And when we remove the endothelium, you can see these are different experiments than up here. In the presence of caspase inhibition, we essentially shut down cell death. We're blocking caspases. We still have a bit of cell death when we remove the endothelium, but we are significantly attenuating it when we shut down caspase activity. And endothelial cell death is so low under these conditions that caspase inhibition has no effect. Okay, and so now I alluded to this earlier, but we're going to show is that endothelial cells protect smooth muscle cells from calcium entry. So here we're looking at the calcium entry during 50 minutes of hydrogen peroxide in the presence of the endothelium. When we remove the endothelium, you can see we get much greater calcium entry. Okay, and smooth muscle cell death, again, you've seen this before, but now lower smooth muscle cell death in brain versus muscle. When we remove calcium from the extracellular fluid, we basically eliminate smooth muscle cell death. And the endothelial cells, as we've seen already, are very much resilient, irrespective of extracellular calcium. So most of this is happening, most of the effects of hydrogen peroxide are happening in the smooth muscle. I should also tell you, I didn't mention it earlier, but we are incubating the vessels with Fura-2 in the extracellular fluid. And all of the experiments that we've done point to the dye being taken up in the, primarily in the smooth muscle cells. Okay, so there's very little Furin-2 that actually gets into the endothelium. That may be one of the reasons. Well, no, that's not true. We've looked at endothelial cell calcium, as you saw in Matt's paper, and when there is not a protective layer of smooth muscle around the endothelium, then hydrogen peroxide also induces endothelial cell death. So while I'm telling you that the endothelium protects smooth muscle cells, the reciprocal is also true, and that is that smooth muscle cells are protecting the endothelium. It works both ways. All right, and so then what is the mechanism? So ruthenium red is a non-selective inhibitor of TRP channels. These are transient receptor potential channels. There's been a huge explosion in the literature on these. Scott Early and his group wrote a very nice review in Physiological Reviews a few years ago, and their lab has been one of the leaders in looking at TRP channels, more so in the context of regulation of channel function. Here what we're showing, we'll just repeat this. Okay, in the with endothelial cells intact, if we then add ruthenium red to block all TRP channels, block calcium entry, smooth muscle cell death goes to 0. Okay, if we denude the endothelium, you've seen this already, more endothelial cell death. If we block TRP channels, we basically shut it down, and again, it's low already in the endothelial cells. If we look at calcium entry, okay, here's calcium entry in the presence of the endothelium, in the absence of the endothelium. And under both conditions, if we add ruthenium red to block TRP channels, again, we pretty much shut down cell death, or rather shut down calcium entry. Okay, and I don't have time to show you all the data, but if you're interested, you can read Chuck's paper, and you'll see that the conclusions, but inhibiting TRPC3 basically shuts down cell death, and inhibiting TRPV4 attenuates it markedly. So we have localized the effects of hydrogen peroxide to these two ion channels. And again, you'll have to read the rest of Chuck's paper to really get into the nitty-gritty of those details. Okay, so in terms of summarizing the effects of vascular cell resilience to hydrogen peroxide, endothelial cells are more resilient than smooth muscle, females more so than males. You saw this earlier, but I want to leave you with a message in this part of the talk that the vessels in the brain are more resilient than those in skeletal muscle. All right, take a minute and breathe. I'm going to change topics now and move slightly, and I'm going to move now to skeletal muscle injury. And what we're going to be dealing with now is injury and regeneration in skeletal muscle. Okay, and I'm going to start. Any of you that are involved in any kind of activity have probably experienced a local muscle pull or tear where you have local trauma. Okay, and what you've done is you've damaged the skeletal muscle fibers, and you're going to induce a local inflammatory response and a mild level of muscle regeneration. A very common surgical procedure is a muscle flap. In this case, I'm showing an illustration being used for reconstructive breast surgery following mastectomy. In this case, the latissimus dorsi is cut and then moved and used to as a transplant, basically to reconstruct portion of the chest. This is a much more severe injury. And then probably the ultimate form of injury, and this is a SIM image, is bad luck with heavy farm machinery or in combat where you can actually have a limb blown off, is volumetric muscle loss. So this is a progressive increase in the severity of injury. I want to suggest that the events going on at the cellular level are not all that different. It's just the magnitude that differs. Here we have a very localized effect. This is an intermediate effect, and this is a major effect. And the relationships that I'm going to be showing you now using mouse models, I think pertain to all of these conditions. The biggest difference is that if you exceed a critical volume of muscle loss, muscle regeneration just doesn't work. You can't regenerate a whole limb. Yes, we're working on it, but we're not there yet. A little bit of background on skeletal muscle ultrastructure. You probably have seen some of this. Epimysium is the outer layer of connective tissue. The perimysium is what surrounds individual muscle fibers, or sorry, fascicles of muscle fibers, and then each individual muscle fiber is surrounded by the endomysium. Okay, and ECM, extracellular matrix, so it's the connective tissue basically that's holding everything together for individual fibers, groups of fibers, and then the muscle as a whole. And we're going to blow this up now and look at the next level. Okay, we blow this up, and the endomysium around the muscle fibers is a scaffold. It contains blood vessels. Okay, and so it's a sheath, it's a scaffold, and the green here is another depiction, if you will, of the endomysium. In this case, the muscle fibers are empty. Okay, and if we blow this up and look more carefully at what's going on in the endomysium, contains a lot of kinds of cells, including fibroblasts and macrophages and adipocytes, but also the endothelial cells and smooth muscle cells of blood vessels, and you can see some RBCs in here as well. Okay, and if we blow that up yet again, here this is the myofiber, this is the sarcolemma, the basement membrane of the muscle fiber, and then the endomysium sitting outside of that. So these are separate layers: basement membrane and the extracellular matrix. Okay. And so injured muscle fibers have a remarkable capacity to regenerate, and that's attributable to satellite cells. And this is a simple figure taken from this review article. Where muscle is injured, it releases a bunch of stuff. I'll just say growth factors. It attracts a bunch of different kinds of cells, inflammatory cells, activates fibroblasts, primarily through signaling through cytokines. Satellite cells are activated. They differentiate into myoblasts, further differentiate into myocytes, which fuse into myofibers. Okay, and the key players here then are satellite cells. So the question then I'm going to address in the rest of the talk, after one or two more introductory slides, is what about the vascular supply? For those of you that are not familiar with satellite cells, these were discovered or first published by Morrow in 1961. Okay, and he showed them in frog and in rat skeletal muscle. This is the inside of a myofiber. You can see myofibrils and mitochondria. Here is the basement membrane. Oh, I'm sorry, the plasma membrane of the muscle fiber. This is the basement membrane of the muscle fiber. The extracellular matrix has been removed here. This big nucleus sitting here is a satellite cell. So these are quiescent, dormant, under resting and healthy conditions. And in response to injury, they become activated and all kinds of stuff happens. A huge amount of literature and a big topic of conversation at the recent satellite cell meeting in July in New Orleans basically says that these satellite cells are actively being suppressed. Okay, so satellite cell quiescence is not a passive process. There is a lot of activity maintaining satellite cells in a quiescent state. And these things are primed. As soon as they become activated, messenger RNA is already sitting there, but it's encapsulated. And the messenger RNA is released, and with a matter of hours, stuff starts happening. Okay, so they're really a dynamic cell population. The preparation we're going to be using is the gluteus maximus muscle. We developed this preparation. Dwayne Jackson is one of the folks in my lab that has used this in the past. Dwayne, if you're out there, I'll just say hello, one of my Canadian colleagues. And we've done some interesting preparation studies with this muscle. The way that we do it in the mouse is so the mouse is anesthetized. We dissect it around its insertion along the spine and pelvis, peel it back away from the animal, irrigate it continuously with a physiological saline solution, put it on the stage of a microscope. And this is what we've done a lot of our work on blood flow regulation using intravital microscopy. So this is just to give you an idea of the preparation that we'll be using here. And let's see, I've got to, let's see, excuse me. I got to get out of pointer mode. And back to arrow mode, otherwise the movie won't play. I practiced this earlier. What I'm showing you now, this is an intravital microscopy image. These are muscle fibers. This is this is a terminal arteriole along here and its parent vessel. And what I'll do is I'll start the movie, and what you're going to see is vasomotion. And as the terminal arteriole dilates, as it dilates, you'll see capillaries become perfused with red blood cells. And as the terminal arteriole constricts, you'll see red blood cell flux stop. Okay, so this is just to show you the power and the beauty, what I call sex and rock and roll for exercise physiology. Again, it's studying smooth muscle, endothelial, muscle fiber, and perivascular sympathetic nerve interaction. All the things we think about as exercise physiologists at the cellular level in the intact animal under living conditions. And excuse my messing with things, but that's the way we're going to do it. What I'm showing you now is some mapping done by Josephine Luke, a very talented undergrad who's now completing her PhD in our program here at Mizzou. And what Josephine did was go in and map terminal arterioles and the capillary networks that they supply. Each one of these colors is a different microvascular unit, defined as a terminal arteriole and the group of capillaries that it supplies. So blood flow would come in along the terminal arteriole and then bidirectionally be distributed along the groups of capillaries. And I'm just showing you this to illustrate how beautiful the organization is of the microcirculation in healthy control muscle. Okay, so there are multiple models of skeletal muscle injury in an animal, and particularly mice. In this review, you can freeze the muscle, you can inject toxins, and you can use chemicals. Okay, in this case, barium chloride. And when we got started in this area, we did a lot of consultation, and barium chloride was the way to go. What I'll tell you, it doesn't make any difference how you injure the muscle. Once you disrupt the cell membrane, everything else is pretty much the same. The only big difference is that when you freeze, you also kill the satellite cells in this area. So it takes a little bit longer for satellite cells to migrate and regenerate. But otherwise, the events are pretty much the same. The beauty of barium chloride is that it's cheap and that it's highly reproducible. And the way that we do the injury in the gluteus maximus muscle is we make a cut through the skin. We inject 1.2% barium chloride, about 50 microliters into the muscle, sew the animal back up, and then wait for things to happen. Okay, and again, I've got to do the escape to show you the movie. This is another intravital image. These are muscle fibers. There's a couple of fat cells. This is a venule. And what I want to show you what happens about an hour after we inject barium chloride. And what you can see is a massive influx of leukocytes, neutrophils, flowing along venules. And the neutrophils then will undergo diapedesis, move out into the tissue, release cytokines to attract macrophages, and that initiates the whole degeneration process. Okay, and so 3 days post-injury, you'll see this abbreviation, DPI. There's a few surviving myofibers. CD45 is a non-selective marker for inflammatory cells. Nuclei again are stained with, in this case, with DAPI. So the nuclei are, and what you see is this massive infiltration of inflammatory cells. If you look in the contralateral muscle, basically everything's okay. The nuclei are all located along the periphery of myofibers. There's a few stray nuclei in the tissue, but basically to illustrate that the injury is localized to the site of injection. So it's a very powerful way of looking at skeletal muscle regeneration in a highly reproducible manner. Okay, and the first paper to come out with our work in this area was done by Charmaine Fernando. The major emphasis was looking at the recovery of blood flow regulation during regeneration. I'm not going to show you the data on that portion of it, that is the restoration of arterial reactivity, but rather what I want to do is introduce you to what happens. And I know you've seen some of this by earlier presenters, but I want to show you the same thing happens in the mouse gluteus muscle, mouse gluteus maximus muscle. We undergo a stage of degeneration followed by regeneration and maturation and functional recovery. Okay, and so here we're staining for laminin, which is again the basement membrane. The nuclei are stained with DAPI and embryonic myosin heavy chain. Under control conditions, 0 DPI, no injury, all you see is laminin and the myonuclei there. And then notice where the myonuclei are, they're at the periphery. There's no embryonic myosin. I showed you before an inflammatory response at 3 days. That begins to subside, and by 5 days, we have the initial stages of muscle regeneration. And what you see here are these groups of regenerative muscle fibers expressing embryonic myosin heavy chain. And I know that you've seen in earlier talks that the myonuclei of regenerating fibers are located centrally. So you see that in the regenerating fibers. At 10 days post-injury, there's very little embryonic myosin heavy chain. You're at the later stages of regeneration. Again, central nuclei. Sometimes there's more than one. In this case, there are 3. Okay, all of the regenerating fibers have central nuclei. By 10 days, we're almost done with the embryonic myosin heavy chain. By 21 days post-injury, you see regenerated muscle fibers with central nuclei. They are almost back to the size of the normal control muscles. Okay, I want you to remember this 21-day post-injury time point for recovery, if you will, of muscle structure. I'm going to introduce you to a technique used in the lab to evaluate perfusion. Right behind the eye is the retroorbital sinus. And if any of you have ever tried to cannulate an artery or vein, either in the tail or elsewhere in the mouse, it can be done but it's not exactly easy. This is easy. You take a 31 gauge needle, however much stuff in your needle. We typically use 200 microliters, inject it into the retroorbital sinus. And this is what you see when you look through the microscope. In this case, it's a fluorescein isothiocyanate conjugate dextran, 70 kilodaltons to approximate the size of albumin. And what you see, the larger vessels are venules, the smaller ones are arterioles, and you can barely see capillaries at this magnification. But it's a way of evaluating microvascular perfusion in the anesthetized mouse. Okay, and so using this technique, and this now comes back to Charmaine's paper that I alluded to earlier, 0 days post-injury, these are what capillaries look like under control conditions. 1 day post-injury, we basically have completely disrupted capillary perfusion. There's a couple of surviving vessels, but capillaries are pretty much obliterated. By 5 days post-injury, we have this phenomenal angiogenic response, and we see hypervascularity. So impaired perfusion at 1 day post-injury with restoration of perfusion by 5 days post-injury. Now we're going to go to an alternative approach using a reporter mouse. And these are mice that we've bred using a endothelial cell specific, that's a CDH5 is DE cadherin. So using an endothelial cell specific Cre recombinase and breeding it with a Rosa 26 MTMG mouse. This is membrane, tomato membrane GFP. And so this is a ubiquitous reporter. Everything in this mouse is normally red. So here's normally the way it normally works. Promiscuous locus, all cells containing the construct, where basically all cells in the mouse are going to have membrane tomato. They're going to be red. We administer tamoxifen using an endothelial cell specific Cre recombinase. The membrane tomato is floxed. This is taken out. We turn on the expression of green fluorescent protein in the membrane. And so only the cells that have the Cre recombinase are going to be green. And in this case, it is the endothelium. And so when you look at a control uninjured mouse, this is what the capillaries look like when you use fluorescence imaging. The endothelial cells are green and everything else is red. And I'm going to show you a few slides now using this model. Okay, 0 days post-injury is pretty much like what you saw when we're doing perfusion, normal capillaries. 1 day post-injury, the capillaries are severely fragmented, and we think these little green dots are little bits of membrane that are still floating around. But you can see we've wiped out 90% of the capillaries here 1 day post-injury. By 3 days post-injury, we are starting to see endothelial cell sprouting. You can see that here, beginning to see capillary regeneration. They are irregular in how they are generating. By 5 days post-injury, you've already seen we've had perfusion restored. The capillaries are, if anything, slightly dilated. The regenerated capillaries, as I just said, are tortuous and dilated, and they are also leaky. What I'm not going to show you data for, but it's in a paper that we just published, is that the capillary fragmentation does not occur as a direct effect of barium chloride, but rather it is a consequence of muscle fiber damage. You're activating proteolysis with damage to muscle fibers. All the bad guys are released out in the tissue, and that's what breaks down the endothelium. And it's in a paper that I will show you in just a moment. Okay, these pictures are unpublished, but I'm happy to share them with you. 3 days post-injury, this is what it looks like. We have endothelial cells growing in, and you can actually see it looks like they're growing in along the muscle fibers. Okay, capillary sprouting. And this is just a pretty picture of how the endothelial cells are growing back. Okay, and you can see many of them are adopting an orientation along the myofiber. And I'm going to blow up this next image and show it to you here. And I'm going to tell you that myogenesis occurs during the angiogenesis, but I'm also going to tell you that angiogenesis occurs during myogenesis. It looks like both are happening simultaneously. And if you look, here is an emptied muscle fiber. The basal lamina persists. And you can see the myoblasts fusing now to form a new myofiber. This is early stage, 3 days post-injury. And you can see the endothelial cells, angiogenesis proceeding along the muscle fiber. In this case, the muscle fibers haven't started regenerating, but you can see the axial orientation of the capillaries. All right, and I'm just pointing now to the myoblasts fusing along regenerating capillaries. So again, this was a paper that came out earlier this year, and Nicole Jacobson is the first author on it. And what her work shows, you've seen some of the images already, is that the capillaries are disrupted, and when the microvascular networks regenerate, they are disorganized. And again, you've seen evidence of that. So I'm just going to show you the summary. 1 day post-injury, disrupts myofibers, fragmentation. Interestingly, the arterioles are spared. It's only the capillaries that are degenerating. So that suggests again that the smooth muscle cells may be in fact protecting the endothelium. 5 days post-injury, everything is dilated, the capillaries are enlarged. Okay, you see there is initial proliferation of terminal arterioles as the capillaries proliferate. But by 21 days post-injury, the capillaries are reorganizing into microvascular units as the nascent myofibers mature. Remember that 21-day time point. Here it is. So what I'm telling you is that under the conditions of these experiments, myofibers and microvessels are regenerating in parallel, and they both mature and are pretty much restoring their structure by 21 days post-injury. They still have a little ways to go, but they're almost there. Those are the key time points. I want to call your attention. I've got a couple more slides and I'll finish, but I really want to show you our latest and unpublished work. Basal lamina. This is a paper that was 1972, the scaffold for orderly cell replacement. Observations on regeneration of injured skeletal muscle fibers and capillaries. Classic paper. This is from their paper. We studied the reconstruction of rabbit and rat skeletal muscles after injury with freezing, ischemia, or autografting. As I said earlier, each injury produces complete necrosis of cells. The basal lamina remains intact and maintains a map of the spatial relationships between muscle fibers and capillaries. Repopulation of the defect with new cells occurs primarily along the old basal lamina, aided by the ability of each category of regenerating cells to grow along the cell supporting surface of its own basal lamina. So each cell type, the basal lamina, has guidance cues that tells which cells to grow where. And you've seen evidence of that from the data that I just showed you. So in the preceding experiments, the extracellular matrix, the basal lamina, persists. These scaffolds provide guidance cues for concomitant myofiber and microvessel regeneration. A burning question for me is, does angiogenesis precede myogenesis? And so we needed an alternative approach. And Aaron Morton came up with this idea, and I'll introduce him at the end of the talk. What he did was to create a 2 millimeter void with a punch biopsy. Here's the gluteus maximus muscle. He made a punch in the muscle. I showed you the perfusion experiments earlier. This is what it looks like just after we do the punch. There's nothing here, it's empty. 1 day post-injury, this is the 2 millimeter injury here. We're staining for serious red. What we're showing you now is collagen being secreted by fibroblasts. 1 day post-injury, it's almost healed the wound. There's a bit more to go here, about 500 microns or less still has to be filled. Here we're staining for inflammatory cells. This is the injury here. This is a line scanning confocal. Here we're showing inflammation by PDGFR alpha cells. These are FAPs, fibroadipogenic precursor cells. And here we're showing, now we're staining for CD31, which is an endothelial marker. And you'll see the capillaries are basically intact, except for the muscle's been injured. And when we these images, you see whole lots of stuff going on. Inflammatory cells and fibroblasts are in here right away, getting ready to do the big thing. And what is the big thing? When we have a void, it takes a bit longer to start seeing sprouting angiogenesis. These are intact muscle fibers. This is where the injury occurred. By 7 days post-injury, you're seeing endothelial cells sprouting into the toward the center of the wound. By 10 days post-injury, these networks are becoming more complicated. And there's a secondary kind of angiogenesis going on. That's intussusceptive angiogenesis. Okay, and that's basically dividing the cells. So you see division, longitudinal division of capillaries into 2 daughter vessels. Basically, a pillar forms in the center of the vessel and then elongates and divides the vessel in 2. And again, I just want to show you how beautiful these regenerating microvascular networks are. Okay. And so now if we step back and look at under lower power using confocal, 7 days post-injury, the initial stages of angiogenesis. 14 days post-injury, the wound is still devoid of myofibers, but if you look closely, what you'll see is that the vessels have grown all the way across. And it's not until 21 days you see myofibers spanning the wound. And what I'm showing you then is under these conditions where there is a void and we've removed all of the guidance cues, angiogenesis precedes myogenesis by a full week. Okay, and in some places where the muscle fibers don't grow back, we get a deposition of adipocytes. You can't see the adipocytes here, but what you see is that the microvasculature remodels in that area in accord with the morphology. They're little round balls in accord with the morphology of the adipocytes. So the ultrastructure of regenerated myofibers and capillaries is disorganized. Look at this. And I think what's happened is because we have removed the basal lamina, but they no longer know how to reorganize the way that we saw when the basal lamina persisted, as we saw in the earlier model. What I'll tell you is all of this work has been, is now, it's in bioarchives. It's available in the public domain. It's not yet peer reviewed. It's been submitted. We're waiting for our viewers' comments, but I'm happy to share these images with you because, as I say, they are in the public domain. So the summary then is a provisional extracellular matrix filled with immune cells and fibroblasts is generated across the wound within the first day. Regenerating microvessels begin infiltrating the wound matrix by 7 days post-injury and traverse the wound by 14 days. The wound is devoid of myofibers at 7 days. Regeneration of myofibers appears by 2 weeks, 14 days, and it takes 21 days for them to bridge the wound. So there's a week lag. So there's a week delay. Angiogenesis precedes myogenesis. Following skeletal muscle injury in which all tissue components are removed, but by about a week delay. Regeneration of microvessels prior to myofibers suggests that a vascular supply supports the regeneration of myofibers. Kind of makes sense. And that the regeneration of myofibers and microvessels in the absence of pre-existing guidance cues affects the restoration of muscle ultrastructure. It doesn't reorganize the way it should. So my co-investigators in this, Charmaine was a very talented PhD student who developed the injury model in the gluteus maximus muscle. Nikki's work is on reorganization of microvascular networks. Aaron, among many other things, developed the punch model that you saw. Chuck was the one who took the reoxygen species cell death story from muscle into brain and is now pursuing that on his own. And Dee Cornelison is my collaborator here in Missouri. She is the muscle stem cell biologist with expertise in muscle generation. So I will stop there and leave you with a pretty fall picture of life along the Katy Trail in Missouri. Thank you very much for your attention.
Steve, fantastic. Thank you very much. That's great. I love your imaging as always. Whenever I see you talk, you always have some great videos and great images to show us, and no exception here today.
Thank you. Let me just say, I said this earlier, it is so strange talking to just a video monitor. So I appreciate everybody hanging with me as I stumble through this. It's just very
no stumbling. You did fine. Everything was fine. Everything was just fine. So folks, as you remember, maybe we take questions through the Q&A option, not through the chat. So if you have some questions, please go ahead and use the Q&A option here in the, and I'm sure it is 4 o'clock, but we'll take some time for some questions, please. Let me start, Steve, by just, I guess the easy question is the smooth muscle cell death in the first part of your talk, the difference between males and females seemed pretty striking. You know, I guess the easy question is why is there a sex difference in that?
Yeah, and so I can throw out any. The first thing everybody wants to do is point to estrogen, and the bottom line is I don't know.
Yeah.
Okay, we haven't really pursued that. It's just been a very consistent observation, and for some reason, being female is protective. When you look at older animals, that difference tends to subside. And so it's again consistent with, if you will, menopause and the loss of the estrogen. But I really don't have a mechanism, but although it has been a consistent finding that the vessels of females are protected to a greater extent from loss than that of males. I wish I could give you an explanation, but we have not pursued the mechanism on that.
That's okay. The lower cell death in the aged animal, in the aged cells, that's because of lower calcium influx in response to hydrogen peroxide. And
we see this, we see the same thing in females as well, and that is there's a lower calcium influx in the vessels from females compared to males.
And that's nitric oxide sensitive. Is that true? Is nitric oxide synthase sensitive? You block it with L-name and the calcium level entry goes down. Is
Yes,
that correct?
and I think it's not a direct, although as you know, NO is a free radical in and of itself. I think it's primarily combining with superoxide to form peroxynitrite. That is the mechanism of action of nitric oxide being lethal to cells.
Mhm. Okay,
So what I can say is we've exposed vessels to NO for long periods of time. In and of itself, we don't see cell death. Okay, I really do think that in the present, with hydrogen peroxide, it's the generation of peroxynitrite and leading to generation. So it's the generation of superoxide leading to the production of peroxynitrite that causes cell death.
okay, yeah. I'm just looking at Tara Haas's question here in the Q&A, and I guess hi, Steve, great to hear your work. Curious about your male-female data, and she's asking the same as me. Does it suggest that females make less nitric oxide than male endothelial cells do?
I like the question. It's probably out there in the literature. People have used nitric oxide probes such as DAF to look at NO accumulation. And to be honest with you, I usually try and avoid nitric oxide studies because everybody else does it. And I don't mean that in any kind of negative way. But in this day and age, because of the prevalence of NO in the studies, we incorporate it, at least in some of the control experiments. So my guess is you would find differences in NO production between sexes, but I don't really know. If NO production were constant or not different between sexes, and you had a differential effect of hydrogen peroxide on inducing superoxide production, then you don't need a difference in NO. It could be explained by the differential effect of hydrogen peroxide on generating superoxide. That would be how I would think the story would turn out.
Yeah.
Okay.
Well, you and Tara can talk about that in a few minutes again. I just want to ask about mitochondria. It wasn't surprising to me to see the cytochrome C in the cytoplasm in terms of when you incubated with H2O2, because H2O2 is a massive fragmenter of mitochondria, so leads to mitochondrial fragmentation, and doesn't surprise me to see that cytochrome C release there. Were you surprised by that, or I guess that's what it does, right?
It was confirmatory. It hadn't been done in these vessels, and we had the tools to look at it, and it made sense to us when we saw these big increases in calcium. I mean, this is something that I was fascinated with as a grad student, and actually started reading about calcium phosphate precipitation in mitochondria decades ago. But it seemed like an obvious outcome, and so we did the experiments. And as you saw in the images, the mitochondria are fragmented. The cytochrome C leaks out. Caspase 3 and 9 are activated. And as you know, both of those are integral to intrinsic apoptosis. So it was confirmatory, but it defined the mechanism of cell death.
Yeah. I was really impressed with your post-injury angiogenesis. I mean, that looks like a wonderful model for studying molecular basis and morphological basis of angiogenesis just post-injury. It's so dramatic, isn't that true?
Thank you for opening the door on that one. This is exactly the grant that I'm writing now. And in all of the model, if you look through the literature, and I'll wave the flag because it's Aaron and Nikki that did these experiments. All of the models of muscle injury, when you look carefully at the data, you cannot distinguish angiogenesis from myogenesis. And in fact, a couple of labs have pointed out the crosstalk between endothelium and smooth muscle during muscle regeneration. And as studies have gotten more sophisticated, people have recognized an integral role, not only for macrophages, but also for FAPs, the fibroadipogenic progenitor cells, fibroblasts. So what you'll see in the literature is crosstalk between smooth muscle and endothelium being regulated or modulated by the immune response and by fibroblasts. And that's the start right now. And I'm showing you examples of both of those. And so the obvious questions, and there are genetic models and we're working on those, is if we can manipulate the availability of either FAPs or immune cells, how do we affect the ensuing angiogenic response? And there's a couple more players in here that I'm not going to get into because we're not far enough along the way, but I wanted to show you enough, shall we say, complementary evidence for the role of these other cells. If you go to the literature now, you'll see in recent studies in the last 3 to 5 years, as I've already said, really integral roles for the immune response and fibroblasts. One of the things the Animal Care and Use Committee had us do early on is they were worried about causing pain and inflammation in mice when we did the Baron-Croyd injury, and they had us inject the animals with an anti-inflammatory agent. Guess what? No more muscle regeneration. Okay, we block degeneration and regeneration, so we block the effect of inflammatory cells or of inflammation we blocked regeneration by blocking inflammation, basically. And so we made the ACUC happy, but we then said we can't do the experiments that we need to do. And just so everybody knows, when you give this stuff, as I said already, it's localized. The animals wake up, and anybody who's worked with mice knows how resilient they are. Within 24 hours, they're exhibiting perfect behavior. It takes a few hours for the anesthetic to wear off. You keep them warm, monitor them, make sure they don't do anything bad. But you could put them right back in the cage with their littermates, and they're fine. And we've done many studies to define the time points that I've shown you. And so we think the key time points are not only control, but 3 days, the pro-degenerative inflammatory response has begun to subside, and it converts to a pro-regenerative inflammatory response. M1 convert to M2 in a general sense, macrophages, and probably Jim Tidball, more than anybody else, has published papers on the immune response to inflammation. So the inflammatory response and FAPs, the fibroadipogenic cells, do the same thing. And that is, early on they proliferate, but at about day 3, they start regressing as the inflammatory response changes to a pro-healing. If the FAPs don't regress, you wind up with fibrosis and scarring. So the appearance and disappearance of FAPs is integral to both degeneration and regeneration. And I'm still trying to wrap my mind around all of this literature, but that's kind of where things are.
Yeah, I'm just very impressed. That model is, I don't know why NIH is not thinking about it the same way I am. I'd be funding that in an instant in terms of looking at angiogenesis and the relationship between angiogenesis and myogenesis.
I'm going back in, but Dave, as we talked about earlier, I think it's a matter of finding the right study section, right? And when things have gone to a muscle study section, I think they're not quite sure what to do with the vessel stuff. Yeah. And when it's gone to a vascular stuff or a vascular study section, I'm not quite sure that they have a handle on what the muscle folks are doing, or the muscle side of things are doing. So I'll keep going back in until we knock down the wall. And in the meantime, we've been fortunate enough to have enough resources to do the work that I've shown you. And with any luck, this paper that's now in review in bioarchives will come out, and we've got a couple more things in the hopper. And all we can do is show the world that we can do this work with a new twist. And please send money.
Yep, you bet. Okay, I'm seeing no more questions in the Q&A, and so thank you for stay with me here, Steve. I want to thank you very much for spending your last hour with us and telling us a great story. And thank you, folks, for listening. And that's the end of our seminar for today. We'll see you next time. Steve, again, stay with me for a moment, okay? Please.
Thanks, everybody. Thanks for your attention. Feel free to email me if you have any questions. I'll do my best to respond.
Right. Okay, hang on, Steve.
Let's see. We can stop the share here. Let's see. Stop share. There we go. All right, I got Dave. I see 2 people here, Matt and Jen. And now it's just you and me.
Okay, and we've got Tara still. Let's see, let me see if I can make Tara see if I can get Tara in here. Okay. So, okay, promote to panelist. Tara, you there?
I can see Tara's name on the screen.
Yeah.
Now she's muted.
All right.
No, there she is. There she
There,
is.
yeah.
Okay, we're good. We're
I
good.
think you can stop recording us though.
Yeah, there's a lot of buttons here, Tara.